Hi everyone, welcome to our weekly AP Bio Chat. Today we were going to do sort of a review of gene regulation, so talking about operons. However, a few questions came in via email and also via Instagram, so we're going to start there. And then I think we're going to wrap up the session by going over last week's biotech free response question, which was about a gel electrophoresis restriction digest experiment. So just so you know, my name is Lina, and you can feel free to email me if you have any questions about today's chat or about any of our previous chats. All right, so I'm seeing in the chat right now that there is a question, does triplet reading come from 3' to 5'? So Arena, in terms of triplet reading, we're going to be reading the coding strand. So the coding strand is going to be used to generate an RNA transcript, and then that mRNA transcript is going to be what is read in terms of the codons. Okay. All right, so let's go ahead and just get started. So the DNALC is doing all sorts of live events. You can check out our schedule of programming by going to this website. There are live labs and video shorts explaining different DNA concepts that you might find useful. So feel free to check out our website. You can also feel free to check out our different social media platforms. So obviously you're on our YouTube channel, but you can check us out on Facebook, Twitter, or Instagram to keep up with what we're doing to support students and teachers in the midst of our current situation. Just a reminder for AP Bio students, we're focusing on the first 6 units, and there will be 2 examinations this year, first on May 18th and second on June 3rd. The exam format is considerably changed, so there will be no multiple choice questions. It's going to be entirely free response based. There will be one 25-minute long and one 15-minute long free response question. And again, feel free to submit questions via email or to our Instagram page. So I'm going to start with a couple of questions we received on our Instagram page. So the first is to explain Mendels Laws. So the 2 that you're going to need to know for the AP exam, and you should know what these are and be able to explain them in your own words, but also know experimentally what observations Mendel made that allowed him to conclude these things. So the first is called the law of segregation. And if we just think about what the word segregation means, it means separate, right? So the law of segregation basically says that alleles are going to separate when gametes are formed, and they're going to do this randomly. I'm going to talk a little bit more about the law of segregation with a diagram that I think is useful. And then the second law says, so it's called the law of independent assortment. And if we break down like what this sounds like, independent just means on your own, right? You're not being caused to do something by someone else. You're doing things on your own. And then assortment just means, sorting into groups, right? So the idea of the law of independent assortment is that genes, different genes, are going to assort independently of one another. In other words, if one pea plant got a gene for green seeds, that plant would not be any more or less likely to inherit a gene for being tall or short, right? So basically the idea that genes aren't linked. And then importantly, these laws are not always observed. So we now know that there are genes that are linked because they're so close to each other on the chromosome. And so it's important to know that, back in the day when Mendel was doing these experiments, we didn't have a lot of the technology or capability to understand what's going on a molecular level. And so these laws are not always observed. So the law of segregation is basically going to say that only one of the 2 gene copies or alleles goes into making each gamete. So if we look at this diagram here, This is coming from the observation. You probably talked about this in your class where Mendel took true breeding purple plants and bred them with true breeding white plants. And the idea of inheritance back in the day was that offspring are kind of like a blended inheritance situation where, kids kind of look a little bit like their mom and their dad. So the expectation before Mendel did these experiments was that the flowers would end up being like a light purple, but that didn't happen. They ended up being the same dark purple as the purple parent plants that were true breeding. Um, the weird thing that happened was that when Mendel self-fertilized this F1 generation, he noticed that the white uh, phenotype reappeared in the second filial generation. Um, and so how was this possible, right? Well, Mendel realized that what was going on was not necessarily blending inheritance, but instead he posited that these factors that were going into making, in this example here, the seeds yellow or the seeds green are split apart, right? In other words, they're separating. So because they separate, the F1 gametes are producing um, basically a combination in which the yellow overpowers the green allele. But then when this generation makes its gametes, again, separation of those alleles is happening. So the offspring are not inheriting a combination of big Y and little y. Instead, the gametes are either going to contain the allele for yellow seed pods or green seed pods. And then when you self-fertilize them, you end up with this 3-to-1 ratio. So the law of segregation is coming from this 3-to-1 second filial generation phenotypic ratio. And this mirrors what we know about meiosis, right? So in meiosis, you learn about the separation of those pairs of chromosomes such that each gamete ends up with half the genetic material of the original cell, right? Okay. The law of independent assortment basically says that if you are looking or if you're studying the inheritance of two genes at once, just because you get one gene doesn't make you any more or less likely to get the other type of gene. So in this particular case, just because you're inheriting the dominant form of seed color doesn't mean that you're necessarily going to inherit the dominant form of seed shape, right? And yes, Suraj, so both of these are going to contribute to variation within populations, right? Okay. And so the idea here is that genes are divvied up or assorted separately or independently. And so this was shown by that 9-to-3 to 3-to-1 ratio, right? And so the sort of idea here is that just because you are inheriting a yellow seed color gene doesn't mean that you're also going to have a higher chance of having smooth seeds, right? It's still these genes are independently assorting. It's important for you guys to know that if you were to get a data table, right, and this goes back to the chi-square example that we did a week ago, maybe 2 weeks ago, we looked at ratios that maybe deviated from this 9-to-3 to 3-to-1 phenotypic expectation. And if you do see deviation, then that is evidence, right, depending on your chi-square value, that could be seen as evidence that shows that the inheritance pattern that you're observing is non-Mendelian, right? Okay. So this is not a exhaustive list, but this is a good set of non-Mendelian inheritance patterns that you should be familiar with. So definitely you want to know sex-linked traits, the idea that you can have genes that are linked to the X chromosome and what sort of implications that has for offspring. And then codominance and incomplete dominance are going to be important. So codominance is the form of inheritance where you see both alleles expressed at the same time. So a good example of this would be blood types, right? So you probably remembered learning about those glycoproteins that are expressed on the surface of your blood cells. If you are type AB blood, you're going to have a co-expression of the A and the B type proteins. Incomplete dominance is going to be the blending inheritance, right? So you probably learned about, I think they're called 4 o'clock plants. But you probably learned about an example with flowers and how, like, when you combine a red and a white allele, rather than, one completely being dominant over the other, you have incomplete dominance. In other words, the red does seem to overpower it, but you do see the white trickling through. And so you end up with like pink flowers in the heterozygote. Um, and generally speaking, so again, thinking about the free response questions, I can totally see them giving you an experimental setup, right? Where they're observing offspring of rabbits or, I don't know, turtles or flowers, right? Flowers might be a little bit too easy. They might try and like psychologically panic you and give you some sort of animal or bug. But if you see deviation away from these 3-to-1 and 9-to-3 to 3-to-1 ratios, you should be on the lookout for non-Mendelian inheritance. So Raj, I don't have a ton of time to do ABO blood groups, but if we have time at the end, I will revisit because I am trying to make sure that we can get this done in half an hour. Okay, so another question that someone had on our Instagram is, do you know if there's a difference in difficulty between the May and June AP tests? In theory, there shouldn't be, right? They should, make sure that both of these are psychometrically fair, and make sure that neither is more difficult than the other. However, right, there's a level of subjectiveness that goes into whether or not you think an exam is hard, right? If you are really good at genetics and you get a genetics question, there is sort of an element of luck involved, right? If you're really good at genetics but sucky at, I don't know, the cell cycle, and you get a free response question on cell cycle, that's just like really unlucky, right? I do think the element of luck might be a little bit more strong for this current AP format, just because there are only 2 questions and there are 6 units that they're testing you on, and each of those 6 units has so many topics, right? So I think a really good study strategy for you is to, and I think I've said this before, go through those 6 units, create like a little organizational chart for yourself, write down key concepts, key terms, and really visit those key concepts and key terms that you're weak on, right? I mean, that's always generally good study advice, but in particular for these upcoming APs where I think the element of luck is a little bit more strong, how do you like fight back against just getting lucky? Well, you make sure that you're good at everything, right? Which is a lot easier said than done, I suppose. Okay. And then the last sort of question that you guys had, I'm going to do this like a very minimalistic view of cell cycle regulation. And the reason for that is because what they're expecting you to know are the nuts and bolts. They're not expecting you to memorize the names of enzymes or cyclins or cyclin-dependent kinases. You don't have to memorize any of that, but you should be familiar with the idea that they exist. And you should be familiar with the idea that tinkering with these sort of checkpoints can have devastating consequences for the organism that they happen in. So cell cycle regulation broadly, for those of you who like totally forget, remember the cell cycle is how we make more cells, right? And it's a cycle because it happens over and over again. And different cells go through the cell cycle at different speeds, right? So you probably learned that, for example, your skin cells and the cells that line your digestive organs, so like your intestines, have a much faster rate of cell cycle replication than, say, your eye cells do, right? And this is why, your vision tends to degrade over time, because that cell cycle is not happening at a very fast rate with your cells in your eyes, right? And it makes sense that you would need to replenish your gut lining pretty frequently, right? Because you're always shoving food down there and things are churning and moving. So there's a lot of like chaos happening inside your intestines. So you've got to replenish those cells. In any case, how do our tissues make sure that we're going through the cell cycle at a pace that allows our organs to function? Well, they utilize these things called checkpoints. And the checkpoints are kind of like referees. So they tell your cell, okay, you're good to go from G to S phase, right? Or they tell your cell, okay, you're good to go from S phase, you're ready for M phase, and so on and so forth. And the referees are basically these things called cyclins, right? So cycle meaning, like cell cycle, and then this INS just indicates that it's a protein. And these cyclins have partners, so they don't do this alone. They have to bind to these enzymes called cyclin-dependent kinases. And cyclin-dependent kinases are simply enzymes who have specific roles in turning on or off proteins that allow the different steps of the cell cycle to happen. You should know that if you tinker with these checkpoints, you can end up with out of control cell cycles. In other words, cancer, right? So cancer is the inability for your tissues to regulate their cell cycle properly. Or on the flip side, if you can end up with programmed cell death or apoptosis. So again, I've sort of highlighted it here in italics. You don't need to memorize this stuff. What you need is a firm understanding of the basic ideas and the basic molecular players involved. I think that if they do ask you anything about cell cycle regulation, they will surely show you graphs. So I'm going to go over a couple with you and like what they mean. So the basic idea is that there isn't just one type of cyclin, right? You can imagine if there were only one cyclin type, you wouldn't have specificity for each of the phases of the cell cycle. In other words, like you don't have one light switch for all of the lights of your house. That would be highly energy inefficient, right? Instead, you have a light switch for your bathroom. So when you go into your bathroom, you can turn that light on. And when you leave your bathroom, you can turn it off, right? Same sort of thing applies here. We have multiple light switches for the multiple phases of the cell cycle. So here is a little graph, and all this graph is telling us is that these cell cycle cyclins are going to have high concentrations right before the cell enters the phase that it needs to enter. So here you can see, and we'll talk about G1 cyclin in a bit because it's the outlier here. But you can see that the S cyclin increases in concentration right before S phase. That makes sense, right? And then it's going to be prevalent up until where? Well, it's going to start to shoot down right before M phase, right? And then the M cyclin concentration increases and then decreases as you go back into G1. So G1 cyclin is required for all. So what you can infer from this is you can infer that G1 cyclin is required for all of the phases of the cell cycle, right, in high concentrations. Okay. And so the third sort of bullet point here is that cyclins have to activate or inactivate target proteins of the cell, but you don't need to memorize any of this. So the key takeaway here is that there are different cyclins for different phases of the cell cycle. So a little bit more about how cyclins work. So they activate these protein enzymes called cyclin-dependent kinases, and you can tell it's an enzyme from this ASE ending, right? So you can think of this as an enzyme that sets off the different reactions to turn on or off the cell cycle step. Again, I like thinking of light switches. And we're going to look at one example of an interaction between a cyclin and a CDK. Involving these things called maturation promoting factors. MPFs, you don't need to know what these are. I'm just giving you an example here, but they are basically a complex that allows the cell to move from S phase into M phase. And so here's a diagram that you probably have seen or that you might see in an exam free response question. So you can sort of see here that as the cyclin concentration is increasing, it's increasing right before the MPF activity increases, and MPF activity increases as cyclin concentration is increasing. Then you notice that it decreases as soon as you leave M phase. So as soon as you go back into the G phase of the next cell cycle, it decreases because you don't need MPF activity during this phase, and it starts to pick up again right before you go into M phase. This is a diagram simply showing you what is happening in terms of the protein interactions. So the cyclin binds to the CDK, generating this thing called an MPF. So these two together are what we call an MPF, or a maturation promoting factor. And then as soon as you're out of M phase, you can see that the cyclin gets broken down. So the CDK is free to reuse again as you're going into M phase during your next cycle. Another example here is just what it might look like for going from G to S phase, right? Rather than having an M cyclin binding a CDK, you might have an S cyclin binding an S CDK, right? So just be familiar with these types of graphs and don't be afraid of them. I feel like students tend to get panicked if they haven't seen a graph before. The fact of the matter is you will probably be seeing a graph that you have never seen before on this exam. You need to do the best that you can to connect the big ideas to the specific features of the graph here. So what we can sort of see here is that S cyclin is increasing right before S phase. It's at high levels during S phase and during G2 phase, and then starts to decrease as M phase starts to happen. And then why is that the case? Well, because during M phase, who do we need to be involved? We need M cyclin. So you can sort of see that the cell prepares for M phase by increasing the amount of M cyclin that it's producing. And then that sort of decreases as you go into your next cell cycle. So again, interpreting graphs, I think, is going to be more important for cell cycle regulation on this exam than memorization is. Okay, so P53 is, again, you don't have to know this, but it's good to know because I think that this is another example that maybe could show up on your exam. They could give you a question about what happens if there's a mutation in P53. So P53, you probably learned in your class, is a really, really important gene, and it's nicknamed the guardian of the genome. So the reason for this is because P53 triggers the production of a CDK inhibitor in case DNA is damaged during G phase. So in other words, the G phase is when your cell is sort of preparing to divide. And if there is some sort of mutation that could end up showing up in future generations of cells, right, you would want to stop that from happening. So P53 is kind of like a traffic cop, right? Making sure that people stop at the red light. So basically what P53 does, it stops the cell cycle from happening if there's something wrong with the DNA. And it's known as a tumor suppressor gene because if P53 is not functioning, what ends up happening is you end up making lots of copies of cells that have this faulty DNA, which can lead to cancer, right? So that could crop up in some sort of free response question. And again, I'm just showing it to you, so if you do see it on the exam, you don't panic. All right, and then last but not least, so we've got about 10 minutes left. I wanted to review the free response question we started last week. So it was about restriction enzymes and gel electrophoresis. We had this setup where there was this map, and it showed restriction sites for two enzymes, and they asked you all of these questions, right? So we're going to just go right into them. For A, all they're asking you to do is explain how gel electrophoresis separates DNA fragments. Remember, we're separating things based on size and charge. DNA is negatively charged, so the DNA is going to move opposite the direction of where the electrical current is applied because electricity is made up of these negatively charged electrons, right? So like charges are repulsed by each other, so the DNA moves away, right? And then in terms of size, we use that traffic analogy where the smaller molecules of DNA or smaller fragments of DNA are able to travel a lot further in the gel than the larger fragments are. So my answer sort of reiterates that, but is more succinct than what I just said out loud. Okay, so that was the easy part. And then B is sort of asking us to interpret what the data would look like. So what I did and what I would do is I would like draw myself a diagram. You'll notice I like moved the arrows down so that I can visualize these chunks of DNA that are going to result, right? So they ask you first to predict what the results will be when you digest the DNA with only enzyme A. So basically what that is telling us to do is it's telling us to figure out like how big the bands are going to be. So when you're doing this digestion, you're making a cut in the DNA here, right, at around 450 base pairs. So this right here is going to be our first fragment, this right here. So this little snippet. This is going to be our first chunk or our first band. And it is about, based on where they drew the line, it is about 450 base pairs. So this first fragment is going to be about 450 base pairs long. I did this exact same thing. So now we're going from about 450 for our second fragment. We're going from about 450 to about 1450. And so that's just pretty easy algebra, right? So 1450 minus 450 gives us our second fragment, which is about 1,000 base pairs long. We have two more cuts here. So we're going to create a little tiny fragment here from 1450 to 1550. That's our third fragment. And so that's about 100 base pairs long. And then our last fragment is going to be from 1550, and they tell us in the problem, so this is the one tricky thing here. They tell us in the problem that the DNA is 5,000 base pairs long, and so this is not to scale, but you can sort of just do some basic algebra here. 5,000 minus 1550 from this cut here would give you about 3450 for your last fragment. So enzyme A, and I like to think of restriction enzymes in terms of crafting. If you cut a piece of paper or a string 3 times, you're going to end up with 4 pieces of string, right? Same thing here. If you cut a piece of DNA at 3 sites, you're going to end up with 4 pieces of DNA. Okay, so that's enzyme A digest. B asks us to figure out what the result will be if you only cut with enzyme B. Remember, B only has one restriction site at around 1350, so we're going to be splitting the DNA into a 1350 base pair long fragment and a 3650 base pair long fragment. Then they ask us what's going to happen if we double digest. So this is where things get maybe a little bit trickier. So again, I would just draw the diagram. I would draw the cut so you can visually see like what the expectation is. And so when we do that, we're doing a cut here. So we end up with that same first fragment as just doing the A digest, right? But because we are now cutting with enzyme B, we're going to be cutting that second band in half, right? Well, not exactly in half, but we're going to be cutting it into pieces. So now this is our first fragment. Now we've got this, we've got the second fragment here that is different than any of the digests we've done before, right? And it runs from about 450 to 1350. So when you do that math, that is about 900 base pairs long. We now have these 2 fragments here, right? So we've got these 2 small chunks. One runs from 1350 to 1450, and the other runs from 1450 to 1550. So they are about the same size. And so they actually are going to move to the same part of the gel. In other words, our gel cannot distinguish between these 2 snippets of DNA because they're the same size, right? And remember, this gel electrophoresis can only discern DNA based on size and charge, right? And so these 2 are actually going to make our third band. They're not going to create distinct bands. They're going to show up on the gel as the same, right? And so our third band will be about 100 base pairs long. And then, this last one is going to be the same as last time. It's going to be 5,000 minus 1550. So when we do the math, we get 3450 base pairs in length. All right. And so that is the double digest results. We're going to end up with 4 cuts. 2 cuts produce similarly sized fragments, and so we expect 4 bands with different DNA with different sizes. All right. The undigested DNA. So going, why is this? Oh, there we go. All right. So the undigested DNA means that no cuts would be made. If no cuts are being made, we're just going to end up with the intact 5,000 DNA base pairs. And then they ask you to predict how the results would be different if a mutation occurred at the restriction site for enzyme B. So if there's a mutation in the DNA right here, enzyme B will no longer be able to cut at that site, right? Because remember, restriction enzymes are specific to specific sequences. So if you change the sequence, this cut is no longer going to happen. So if I'm like sketching out notes and doing a test for myself, I would make a little X here. Or if it's easier for you to visualize, you can like literally just scribble this out because this cut no longer exists, right? And now they're asking you what will happen for each of the digests that we did before. So for A, what would happen if you digested with B? What would happen if you digested with both? And then what would happen if you digested or you did not digest at all? So for A, are our results going to be different, right? Probably not because A doesn't involve the restriction site for B, so it doesn't matter. So we're probably going to get the same result. Let's think about enzyme B. If we digest with enzyme B, it no longer recognizes this site. And we're not adding enzyme A yet, so it'll probably appear undigested, right? As if we didn't put any enzyme in at all. For our double digest, well, our double digest is only going to result in enzyme A cuts because we said that the enzyme B site is no longer recognizing enzyme B, or rather, it's really the reverse, right? Enzyme B is no longer recognizing that restriction site. And then for our undigested, we're not really expecting any change, right? So our answer here would just say that it would not affect enzyme A digest. Enzyme B would appear the same as undigested DNA. The double digested would appear the same as the enzyme A digest, and an undigested sample would be unaffected. All right, let's clear that drawing. Thank you so much for joining us today, guys. So next week we'll be here at the same time. I would like to do gene regulation next week, but if you have questions or have more pressing topics that you want to address, feel free to email me or you can ask questions on our Instagram channel. Thank you so much for joining us, and I hope you guys are having a good time studying. I know that it's kind of stressful to study for this, so feel free to give me any suggestions about holding these sessions, and have a good rest of your day/week. Stay safe. Bye.